So we talked in class about genetically modified organisms and focused on genetically modified plants. And then we talked a little bit about an example of a genetically modified organism that had the potential to get into the food supply, but has not. So we talked about the EnviroPig. And it's considered a GMO because it has a gene from a totally different species, E. coli, cloned into it. So let's talk about some other examples of genetically modified organisms that you may have eaten. So there have been some transgenic fish, and this is a genetically modified salmon. It grows, you can see, much larger and faster than normal salmon. Oops. What they did is they took the growth hormone from a related Chinook salmon and they used the promoter. Remember, we've talked about the importance of promoters because they are regulating the gene expression. So the promoter from the growth hormone was from a fish called ocean pout. And the reason they did this was so that the growth hormone would be expressed year-round. Which means normally a salmon only expresses growth hormone in the spring and summer. These are Atlantic salmon, farmed. And so they grow in 18 months to the size that it goes to market, versus normal salmon take about 30 months. The company that does this is called Aqua Advantage. And it's based in Massachusetts. Let's see if I can spell Massachusetts. And this transgenic salmon was FDA approved in 2015, but we're now going through labeling regulations, and so in fact this transgenic fish is not sold in the US, but it is in the Canadian food supply. I can't spell Canada. So you can find this in Canada. These fish are grown in landlocked tanks, so there's no chance for these fish to breed with wild fish. So they are truly farmed in closed containers. But you can imagine if they're growing almost twice as fast as a normal salmon, that's a great money product producer. So transgenic salmon is an example of a GMO that could potentially one day be in our food supply. Atlantic salmon is farmed and in our food supply already, but it's just not transgenic. They were looking at putting in a gene from this little flounder. It's called a winter flounder. And it has what they call an antifreeze gene, or expresses an antifreeze protein. And this is trying to show you what an antifreeze protein does. And in fact, there are antifreeze proteins, not from this fish, but I'm actually not sure of the source, but helps make your ice cream stay nice and smooth. And what the antifreeze protein is trying to do in these fish is prevent ice crystals from forming. Because when you form ice crystals in cells, it can actually burst the cells. So you want to have something that inhibits the ice crystals, so it blocks ice crystal formation. And so even at cold temperatures, things like your ice cream stay nice and smooth. I think the protein that they use for some reason I think is from seaweed, but I don't know why seaweed would have antifreeze. But anyways, there's something like that in our ice cream. Anyways, the idea was to make transgenic fish. Here's my artist for you. Fish that had this antifreeze protein in it so that you could grow. The goal would be to farm fish, grow fish in more areas of the ocean. So maybe where the temperatures were colder. When I tried to find some updated information, I haven't found much else on it, so I'm not honestly sure if this was actually done or not, but I know this was an idea of making transgenic fish. Triploids, remember we talked about, are not technically, they're genetically modified, but they're not a transgene, they're not transgenic. But what triploids do, so we saw examples of triploids that make watermelon and bananas not really sterile. They do that with fish and aquatic species too, but they also increase the size. Because instead of trying to put energy into reproduction, these triploid oysters, and this is a triploid grass carp, just put all their energy into getting larger. The issue is that this guy was made to eat up the vegetation in this poor lake that was just overrun. Unfortunately, these fish got out into the neighboring streams and decimated all the vegetation there. So one of the issues as we genetically modify, whether it's transgenic or through breeding or other methods in the lab, is we have to be careful if these animals kind of get out of our control. There are triploid trout that are, I guess, actually pretty popular in some fishing tournaments and stocking lakes because they grow nice and big. So they're a great trout to try to catch. So again, genetically modified, humans have done this, would not happen normally, but these are not transgenic, so they're regulated in a totally different way. One that's pretty cool that I'm not sure right now, it's still in use, but I couldn't get a feel for if it's a big producer. Anyways, is the idea of expressing silk. And this is a silkworm. And this is obviously a spider. And the idea is producing this spider silk into goat's milk. Okay. This is called biosteel. Okay. And the goal is that this silk, so here's a spider spinning its silk, would work kind of like Kevlar. So be kind of bulletproof, light, strong, have some elastic give, moisture proof. And so there is a researcher at Utah State that is working with these goats. He's making spider silk in transgenic bacteria as well. He's actually making spider silk in silkworms. But you can imagine the issue with using silkworms or spiders themselves is the capacity, right? You would need lots and lots of spiders or lots and lots of silkworms to form enough silk. And I guess it's really the spider silk. We already have silk for clothing from silkworms, but it's the spider silk that's most interesting for its strength. And he's also expressing it in alfalfa. So he just got another big grant from the U.S. Navy to keep working on this. One of the interesting, instead of maybe making clothing like bulletproof or super strong clothing, they're also looking at making things that would work in water and be sticky. And so they have a picture on his website. You can look right here on his website of a propeller kind of all wrapped up. So maybe different applications, and it being so strong might be able to stop a, say, submarine propeller. So the cool thing is it started in goat's milk, and animal milk is a great high protein source. You can use a promoter that expresses the transgene only in the milk, in the mammary glands, and it's a renewable source, right? You take care of your goats, produce more milk. One of the first, or I guess it's actually the first transgenically produced biopharmaceutical is called Atrin. Atrin is also produced in goat's milk. It's an anticoagulant that's used in surgery. And this is just kind of a schematic of how they've produced this. So again, what we've learned about recombinant DNA, with a special promoter that's only going to promote it in goat's milk. To make a transgenic animal, we'll talk about that in a couple more slides, but you're actually working with embryonic cells, surrogate moms, and as you can see, it's going to take several generations to produce a transgenic animal that produces a good quantity of your biopharmaceutical in the milk. But Atrin is a success story. It's Revo Biologics is the company that has the patent on this now. But again, kind of an interesting idea. This is just another example of the challenges with making transgenic animals. It's a long process. And this is before, you're even going through FDA approval and all of that. So you have to go through the goat life cycle a few times in order to get transgenic goats that are going to be high producers. And right here it's showing you a little bit about clinical trials where you're making sure that what the goat is producing is safe and effective for people. This is a little bit of an old slide. I didn't find a new updated one, but a list of therapeutic proteins produced in the milk of transgenic animals. So we have goats, We have rabbits, which I have a bunny at home, cute little lop-eared, and I just never think of, well, he's a boy, so you obviously wouldn't milk him, but never think about rabbits producing milk. But definitely small, easy to breed, easy to take care of. Transgenic cows, and some more goats. So, transgenic animals is what we call bioreactors. Is a field with a lot of research. Again, the idea is that you just harvest the milk and these animals continuously are producing the product that you want. Fairly easy to extract from this nice liquid milk. And then once they're transgenic, you can continue to breed them and they're going to have that new gene in all the offspring. Okay, so my pretty much last slide is just a little bit about how you make a transgenic animal, because we talked about how to make transgenic plants. So there's really two different methods. Both of them, are talking about you start out with your recombinant DNA. So your restriction digest, or your PCR, your restriction digest, your ligations, putting things into bacteria, growing it up, and then you can take the transgene, so you can take your recombinant DNA and actually micro-inject it into, and the pronucleus is right when things are fusing, egg and sperm. You inject it there, and you implant into a pseudo-pregnant female, so you kind of trick her body into thinking she's pregnant, and you start to get offspring with the transgene. So you have to, it says breed to homozygosity. You may not have 100% of cells that take up this transgene. You have to look at, see where it integrated to make sure it hasn't disturbed something else. So there's a lot, just like with the plants, a lot of screening going on to make sure you have the animal you want to. The other way is to use embryonic stem cells. Oh, it says right there, sorry. Embryonic stem cells. So we talked a little bit about stem cells, how stem cells are these great regenerating cells. And so you can take your transgene and put it into embryonic stem cells. Okay. Grow or add those embryonic stem cells into a blastocyst, so into a little ball of cells already. So you're adding another cell. Put it in the surrogate mom, and you're going to have what we call chimeric mice. So they're going to have some cells with the transgene and some without, right? Because they were already put into this little ball of cells that didn't have the transgene. And so you start breeding them until you have a homozygous transgenic animal. And sorry, what I meant to say for here is you're starting here with this single cell, but remember that, sorry I totally spaced this, should know this from genetics, right? You have 2 of each chromosome, right? So you might get the transgene into one chromosome and not the other, and so that's why you have to breed to homozygosity till you have the transgene in both chromosomes. All right, so this is chimeric, where you actually have mice with 2 different types of, or 2 different sets of DNA, and this is where you probably have the transgene only in one chromosome. The other issue is because we're not good at controlling recombination, like I said, where the transgene is going to go, you have to make sure it doesn't disrupt anything, make sure these guys are healthy, and you have to look at copy number. because it might integrate into the chromosome multiple times, and you may or may not want that. So, similar ideas, everything starting with recombinant DNA, and then you're screening up whole organisms to find the transgenic ones you want. And that was going to lead us to Monday, and I apologize for this lecture coming so late. I kind of spaced it this weekend. We're going to talk about what's the problem. Why is there controversy with genetically modified organisms? And there's definitely pros and there's definitely cons. And so I'm looking forward to our discussion. Thanks.